Assalamu alaikum and welcome back. This is our lecture series on DNA and design. This is our fourth lecture, a very brief introduction to mutations. So we've already gone over the structure of DNA, the functions of DNA in terms of replication, to produce cells with identical DNA and to pass on that DNA to the new generation, as well as translation and transcription of DNA to copy the information in DNA into messenger RNA, which is used to make new proteins. Now, you have doubtless heard of mutations in the DNA. And these mutations have multiple causes. They may be chance events. They can be accelerated or exacerbated by things like high energy photons from X-rays or gamma rays and so forth. So we want to just get a very, very brief idea about some of the more common types of mutations. It's a long and complex subject, but we'll just touch very briefly on some of the basics, because that will become important to us in several contexts later on. And having this information now will be very helpful to us when we begin to talk about evidence of design in DNA. So the first type of mutation, which is the most common, is what is known as a point mutation. And a point mutation basically takes one base, one nucleotide, and changes it, or sometimes there's a deletion or addition of an extra nucleotide. But for now, let's just think about substitution of one base for another. And this, of course, will cause a compensatory or similar change in the complementary strand of DNA. So in this illustration, you see that there's a dotted oval highlighting one base, C, which is hooking to the complementary base G, and as we've already covered, cytosine, represented by C, pairs with guanine. In this particular point mutation, the C, cytosine, gets accidentally changed to a thymine. So C gets changed to T, and then the complementary base, now you know that T pairs with A, so the thymine now will pair with an adenine when that DNA comes to replicate. That's how we get that adenine there. So basically, it is a single point mutation where in this particular case, the C changes to a T, and this, when that DNA gets duplicated, will then cause also a change in the complementary strand. The G that used to pair with the C will now change to an A to pair with that new T. So that is how you should think about a point mutation. Okay, so how significant are these point mutations, which once again are the most frequent type of mutations in DNA? Well, sometimes, in fact, most of the times, not very significant, but other times extremely significant. And to sort of understand that range of possibilities, let us remind ourselves of this slide, which we've seen before. Remember that the information in DNA gets transcribed into RNA. The process of copying the information over into our RNA is called transcription, and then that RNA gets translated into a protein. And we have also a 4-letter alphabet in RNA, almost identical to DNA, except that T in DNA, thymine, is substituted for U in RNA, uracil. And so the 4-letter alphabet with the information coming from DNA makes up 3-letter words named codons, and those 3-letter words basically define amino acids or stand for amino acids that will be linked one to another in the order specified by the information in DNA, copied over or transcribed into RNA, and then translated over into a protein using the information in the individual codons. So if you look at this table of codons and what amino acids they stand for, I would just like you to look at leucine. Leucine is actually coded for by 6 different codons. 2 of them begin with U, UUA and UUG, and 4 of them begin with C, and you can see them there, CUU, CUC, CUA, and CUG. What I would like you to notice is that if, for example, I have a codon such as CUU for leucine, if I have a point mutation in the last base, in the last U of CUU, if that were to mutate and change to a C, then I would have CUC and I would still code for leucine. If it were to change into an A, I would have CUA and I would still code for leucine. And if it were changed to a G, I would have CUG and still code for leucine. So you can see that what happens with point mutations here would most likely be nothing, because 3 of the 4 possible mutations still code for leucine. So I just wanted us to be aware of that dynamic. Okay, keeping in mind what we've just talked about, let's look at a specific example of the different types of point mutations. And again, this sort of thinking or knowledge will become important to us later. Please don't think that it is just useless detail. So in the natural situation, let's say I have no mutation and I have the 3 base pairs T, T, C. When that DNA gets transcribed to messenger RNA, that TTC will correspond to AAG, because remember, T pairs with A and C pairs with G. If you were to go back to our previous table, you would see that AAG, that codon, that 3-letter triplet, stands for the amino acid lysine. Now, let's think about mutations in the last base, in the C of the TTC. If it were to change to TTT, where that C changes to a T, then when it gets transcribed, that would correspond to AAA. And just like we talked about with leucine, both AAG and AAA code for lysine, so nothing would happen, and this would be considered a silent mutation. It would not affect the structure of the protein, and that is known as a silent mutation. Now let's say that I have a mutation not in the last base, but in the first one. So instead of TTC, I change to ATC. The first T gets changed to an A. When that gets transcribed to RNA, that would then become UAG. And that particular codon is what is known as a stop codon. We had talked briefly about stop codons, that the codons can either code for an amino acid or code for stop, means stop synthesizing protein at this point, do not link any more amino acids. So you could see that if I had this sort of mutation, that would be very significant, because if I have, for example, a 200 amino acid protein and I get this sort of mutation in, say, the 10th codon, then all of a sudden, after 10 amino acids, we don't link anymore and we stop. So I can't make a functional protein. And this is known as a nonsense mutation. The last type of point mutation is known as a missense mutation. And so look at now the middle base, the middle T in TTC, and let's say that gets changed to a C. So now I would have TCC. With mRNA, I would have AGG, and the lysine would then be changed to an arginine. Now, so now I substitute one amino acid for another in the protein. Instead of having a lysine for that codon, I would have an arginine. In this particular instance, this is known as a conservative mutation. Why is it a conservative mutation? Because arginine functions sort of like lysine. Both of them are basic amino acids. By basic, I mean basic and acidic. So both of them are basic amino acids. They don't terribly much affect the way the protein chain folds and may make the protein a little bit less efficient, but would probably not have a devastating effect on the protein. And so it is known as a missense mutation, but a conservative missense mutation. If, on the other hand, instead of changing TTC to TCG, I were to change it to now the last column to TGC, where that middle T is changed to a G, then when I transcribe to mRNA, I get ACG, and that ACG will now code for an amino acid called threonine. And this is a very different sort of amino acid. It's a polar molecule, and this will now change the structure of the protein, because when I throw in this polar amino acid and the protein comes to fold, remember we talked about how proteins are made of amino acids, a long chain, and then they fold on themselves. And we will talk more about this issue later, inshallah. But for now, you can see that this change, or you can hopefully believe me, that this change would be more significant than the conservative missense mutation. And so this is known as a non-conservative missense mutation. So you can see the different range of possibilities that point mutations can have on the structure of a protein. Okay, let us just do one example from medicine, just so that you see that these things can sometimes truly be very significant. Look at the little snippet of DNA, and this is part of the DNA which codes for hemoglobin, the hemoglobin molecule. And you know that hemoglobin is found in our red cells, and it is what carries the oxygen. And so it is critical to life. Now, look at the double-stranded DNA. We have GAG on top and CTC on the bottom, because as you remember, G pairs with C and A pairs with T, always. And let us now look at the bottom DNA strand, the CTC. When that gets transcribed to RNA, that will give us an RNA with a codon that reads GAG. GAG is a codon that codes for an amino acid known as glutamine or glutamic acid. And if we have one simple point mutation, instead of CTC in the bottom DNA strand, we change that middle T for an A. So now it reads CAC. When that gets transcribed to RNA, it will become GUG. Let me again remind you, in RNA, there is a 4-letter alphabet that is the same as DNA, except that we have a substitution of U for T. In RNA, we have uracil instead of thiamine. So we said C pairs with G, A pairs with T if we're making a new DNA molecule, but A will pair with U instead of T if we are making RNA from DNA. And that's why the CAC then codes for an RNA that is GUG. Now that codon, GUG, instead of coding for the amino acid glutamine will code for an amino acid called valine. And that single change, one amino acid in a very long protein chain, causes the protein to fold abnormally and to function abnormally, and produces a condition called sickle cell anemia, which is a very, very difficult disease. Because the red cells with this abnormally shaped protein, when you get conditions of low oxygen, the protein has an abnormal fold and it causes the red cells to lose their normal shape. Instead of being a biconcave disc, sort of like a donut without a full hole in the middle, they become sickle shaped, and these sickled cells clump inside the capillaries and they actually stop the blood flow. And people suffer anemia, people suffer severe bone pain, infarctions of the bone, could have infarctions or strokes in the brain, and so on. And sickle cell disease is a very difficult disease, and as you know, it is most prevalent in sub-Saharan Africa, but is also found in African Americans. Now, other significant diseases that are caused by point mutations of the general variety that we've discussed are, for example, a disease called cystic fibrosis, which is from the deletion of 3 bases in a DNA, 3 contiguous bases. And remember, we said point mutations are the change of one base to another, the most easy to visualize, but we could also have deletions of bases. And another very significant disease is Tay-Sachs disease, which also has devastating effects on patients. And again, this is not a lecture in medicine, so I don't need to go through what cystic fibrosis does and what Tay-Sachs syndrome does and so forth. I'm just trying to give you a feel for when these point mutations become significant, that can have devastating consequences. So hopefully that will, give us a sense that, hey, I should take this issue of mutation seriously. And when we come to talk about issues of design in DNA, which we will begin inshallah with the next lecture, now you have a good primer and a good sense of what DNA mutations are. Okay, thank you. Hopefully we'll see you in the next lecture. And this again ends the background material. So if you have made it this far, hopefully from here on out, now we can get into the really interesting stuff, inshallah. Take care and salamu alaikum.